Welcome everybody to this, the third of the Synthace DOE Office Hours webinars, third in our series. Hopefully a few of you are here who've caught some of the early ones, but if not, don't worry. There's always the chance to catch up. Everything gets recorded. We'll get going in a second. Just before we do, just a couple of quick housekeeping notes for you. Yeah, first off, hello, I'm Sid. Some of you may have seen me in previous two webinars. You're fortunate enough that I'm not actually going to be presenting today. I'm just here to help moderate. So I'll be disappearing off stage in a second and just lurking in the chat, trying to help answer any of the questions that you have that come along as we're going. But yes, so this is newish webinar software. We used it the last two and it mostly worked. But yeah, if you do have any technical difficulties, please just send us a private message in your chat box and we'll try and help you out. You should be able to see and hear everything fine. Yeah, so as I said, this is being recorded. We'll send an email as a follow-up with this and links to any relevant resources. And then, yeah, you'll be able to access it on demand on our website. And yeah, so the point of these sessions really is to be like super interactive and have a nice conversation. Yeah, I really do encourage you to add any questions, comments, thoughts, whatever you have into the chat. And hopefully we can get a good sort of dialogue going with the presenters and your fellow attendees. We will leave a bit of space to answer questions from the presenters during the end of the webinar, but absolutely please just put them in whenever they occur to you. And then, yeah, at the very end, most importantly, we're going to have a survey for you. Be brilliant if you could fill it in. Doesn't just help us, of course it does massively help us, but it will also help you because it will mean we'll present better, more relevant, more interesting content to you in the future. Yeah, do please give us any feedback you have in that survey. All right, so we're just about ready. Yeah, before we dive in, I just want to quickly say a word of introduction about our speakers. But yeah, you're extremely fortunate to have two real experts in DOE implementation in Synthace here, in Ross and Nuno, who've been working with DOE for a really long time, 6-10 years, and working with Synthace for a lot of that time. And so they've got a lot of experience, both in general with DOE and with Synthace specifically, across a huge broad range of different clients and different scientific use cases. They also know other things like automation and all sorts of things about biological sciences, which I couldn't possibly go into. So yeah, you're in absolutely safe hands with these guys. So yeah, I'll hand over to them and disappear from the stage. Have fun.
Thank you so much, Sid, for the kind introduction. I'm happy to start and immediately pass it off to Ross. So Ross, this is a question that we get a lot from people who are new to Synthace. So what exactly is the difference between Synthace and other DOE tools that maybe they're more familiar with?
Yeah, brilliant question. Thank you very much, Nuno. As you've alluded to there are a number of different sort of tools out there in the wild that allow you to design and analyze. Some examples being things like Jump and Design Expert, Minitab. You can even go it alone and try and design your own. But maybe just to dive into this a little further and take us back to maybe a familiar slide for those of you who joined us in the first couple of webinars in this sort of office hours series, and just take a look at the DOE process. And from here, we can see where those tools tend to fit in and how we typically see Synthace differs from some of these. So you may remember, for those of you who were in the previous sessions and for those of you who weren't, just a little sort of recap for you. Typically, DOE looks generally something like this, right? So 5-stage process for each DOE within your campaign of DOEs. We're going to start off by identifying the factors and levels that we want to investigate. We're going to select a design that's going to be appropriate for us to investigate those factors, sort of to the depth that we're looking for. We've then got the painful process of going in the lab and actually executing that design. These designs can be complicated. Often every run within the design is almost completely unique and just different from everything else within that design, and executing them can be a pain, and we'll allude to discuss some of that a bit later on. You then got to go through your data, you've got to structure it, you've got to get it all into a sensible format and ultimately build some models and analyze that data, and then make your decisions, learn from the process and go through your next DOE iteration, learning each time as you go about your system. I think it's fair to say that kind of process for anyone who's tried to execute DOEs themselves can often leave you feeling a little bit like this guy. They can get pretty complicated. They can get complicated for a number of different reasons. There are a lot of tools out there to help you through that process to answer the question. But fundamentally, one of the things that we tend to see is that often those tools are focused on solving one or maybe two steps of this process, right? So we have tools like Jump and Design Expert that are really fantastic tools, and I can't fault them at all. They're typically set up for statistically minded people, maybe people who are statisticians, to enable them to choose designs, to go through the process of assessing whether the design type that they've chosen is going to be appropriate for the experiment that they're looking to do. And so they tend to be more focused on solving one of the steps in this DOE process. With things like JUMP, I guess you're also looking at modeling your data and doing some of the statistical analysis as well. But this process can ultimately be very complicated. And what we see at Synthace is that whilst you can often generate a design to run a DOE, translating that design into something that you've actually got to go into the lab and execute can fundamentally be incredibly painful, right? There's so many different considerations and things you have to take into account when planning one of these complicated experiments. Take something simple like calculating all of the concentrations and volumes of all the liquids that you've got to move around to apply DOE to your assay. It's hard and it's error prone, and it's very easy to lose track of samples, to make mistakes. And whilst those tools are great for allowing you to design things, they don't really help you so much with actually executing your experiment, right? So yeah, this process can be very time consuming, it's complicated, and it ultimately requires you to become an expert in that piece of software when maybe that's not something that you have prior experience of, not something that you're looking to become a statistician, and you're forced to by proxy in some ways. And yeah, as I said there, ultimately the planning, the design that you've generated doesn't have any knowledge of how to put that thing into practice. We tend to see that's tricky in the first port of call, but often you start planning these complex experiments, and it's only at that point that you realize that actually the design you've generated maybe doesn't quite work for whatever execution reason in the lab. There's something not quite right with how you set things up. You want to change your mind, you want to change your design, you want to change your process. And so you've got to go back and start replan everything again from scratch, right? And it can be painful, it can be really painful to do. And that's just the designing and planning. And then it comes to actually, you've gone through the pain of running the thing in the lab, and then you've got to get all your data back together, structure it against your design, which is typically randomized, which just makes things a little bit harder. You might have data spread across multiple different files, different formats. You've got to combine all that back together before you can even start going through the modeling and analysis process, which again, time consuming, it's complex. You end up becoming an expert copy and paster, which I don't think anyone really enjoys, let's be honest. It's ultimately just very easy for mistakes to creep in as well. And going back to the question, how does Synthace compare to those tools? Fundamentally, we try to look at all of the DOE process through the lens of a biologist, and we'll touch on this in some of the later questions as we go through. But what we've ultimately set out to do, the mission behind Synthace, if you like, is to put DOE into a scientist's hands in a way that combines the last 4 steps of this process. Unfortunately, we can't help you select what things to test and how to test them. You still have to apply your scientific brain to that part, but we can help you with the rest. So by putting all of those tools in one place and strengthening that connection between the actual running of the experiment and the design that you've selected, and that's where we really see the differentiator. So we do this ultimately by allowing you to generate execution instructions, whether that's for manual liquid handling or something automated. And I know Nuno's going to talk about this one in a second, so I won't steal your thunder too much, Nuno. But yeah, ultimately it's about putting all the tools in one place and enabling scientists to execute complex DOEs.
I really like this slide because even though we present it as the DOE process, really this should be or is the experimental process, right? I think in DOE specifically, you put statistics and the design planning before you actually execute. But I think we all know too well that we should do that to all the experiments that we run, right? Like that at some point you might have learned a hard lesson because you didn't quite plan the experiment as thoroughly or with the statistics in mind before you actually set out to do it. So this really exemplifies the entire experimental journey, and Synthace, plays a big role across these different stages.
Yeah, absolutely spot on, Nuno. And maybe the perfect segue into our next question. So this one's for you, Nuno. So what role does Synthace play in the DOE process? So to maybe dig into that a bit more.
Yeah, so you've alluded to some of the challenges that kind of come with the DOE process. And so I'll highlight them and see how Synthace responds to it, but then also show you a little bit of how it actually looks like, what it actually looks like in the platform. And there's a series of practical challenges, right? So in terms of tooling, we've alluded that traditional tooling that scientists are familiar with doesn't necessarily support DOE. And then you have DOE specific tooling that requires significant expertise that biologists may not have, right? Then there's also the case of, on the execution side, that there's this disconnect between the design and the execution plan, which makes planning and running complex experiments like DOE much more challenging and difficult. And we need to think as well that when we're running DOEs, we're running usually highly combinatorial, complex experiments. And so you're likely to want to use automation to do, and that means there's another layer of software that you need to be familiar with. And so there's many steps of translating this idea of this experiment that you want to run onto an actual plan that you can run, right? And as you said, it's often an iterative process. And so you want to have an idea what it's going to look like before you set out to go into the lab and do it. And so for that, having a sort of visualization of what's going to happen can be really useful. And then finally, you're going to end up with a lot of data that usually doesn't actually have any context of the design that you've specified, right? Your plate reader likely is just going to output your response like absorbance with a well location, but you know that actually in that well, there's a specific combination of liquids that were set at specific target concentrations that are all part of a bigger design, right? And having to align all of this is frankly not the kind of activity that you want a highly skilled and busy scientist to spend time doing. And so what Synthace does to address these things is provide you with a streamlined DOE design tool that is biology focused. So it understands what are the main cases that you're gonna come across with, and also take into account that, yes, you might be specifying a concentration, but that ultimately needs to be translated into a volume that is going to go into a well in a specific robot. That we do this magical step of automatically converting a design into an execution plan. And that is something that really just saves a lot of time and error. And it's wonderful to see the platform work in this way. And then also that we can do, especially with the device that we support, automatic data capturing and structuring. So you don't just have a response, but you have that response specifically aligned with the design that you're implementing, with all the metadata around the execution that you've accrued along the way as well. So how does Synthace does this? So Synthace lets you create a digital representation of your experiment. So you can specify exactly what's going to happen. You can specify your design, and you can actually see what that is going to look like. So you can see a preview of all the plates that you are using in your experiment. You can see how much of each liquid you're going to need. You have an idea of how long the experiment is going to take. And if you notice anything that doesn't quite suit your needs, you can modify it and iterate without ever stepping foot in the lab, right? So then you really only go into the lab when you're ready to execute. And that's when Synthace will automatically translate the plan that you've spent some time thinking about into all the instructions that the device is going to execute. And so you don't actually need to learn how to program these devices. You can use the instructions that are generated by Synthace, and you have this digital representation of it. And then specifically with DOE, we can also structure the data that you generate automatically, and then you can do some model building and analysis within the platform. So in the same software, you can run through the entire DOE process. And so for those perhaps less familiar with the platform, this is our builder. So here is where you can describe what your experiment looks like. And so in this specific case, we are seeding some cells on the Dragonfly, incubating, and then later on the Synthace Flex, we are adding some target cells on top and incubating again. And so you can see how you can define an experiment that is actually across multiple devices, all in a single place, and Synthace will automatically create instructions, match each part of this protocol. And this could be your standard SOP that you're running day in, day out. But when you're looking to optimize this SOP, for example, or a process that you're interested in, you can switch from this build mode into this E mode. And here is where now you can highlight all the parameters that you can modify as part of your DOE, and then build your design. And so you can see how you can apply DOE to your day-to-day processes. So when you're in design mode, then you can specify the factors. So you can see here someone is specifying glucose, glycerol, yeast extract. So suggests that someone is doing a media optimization. And so you specify the liquids here. You also specify the concentrations that you want to vary. So the levels at which you're testing this factor. And once you're happy with the factors that you're including in your experiments, you can switch over to your design choice and design evaluation. So Synthace offers you a few different types of designs, and we'll go into a bit more detail on which ones later on. But also, here's a part that is, again, a bit iterative, because you can evaluate what happens to the power of your design if you change the number of runs. What happens to when you choose a different type of design, to your ability of identifying main effects versus identifying interactions, for example. And you can make a choice on the best design to use to suit your experiment. Once you're happy, you can click here, simulate with design, and this is going to create all the wonderful combinations that satisfy that run matrix that you've just specified, that design. So you don't need to worry about making sure that you're saying this liquid at this level needs to be present with this other liquid at this other level. Synthace will do that for you, and you'll see exactly what the experiment will look like. And you can go back to your design and realize, actually, I still have space. I can maybe create another plate. I can make my design a bit more powerful. It's still within a reasonable execution kind of time frame, for example. And what Synthace will also do is create the instructions for the device, taking the content or the context of your design, but also of the devices that you're using, the labware that you have available, things like residual volumes in plates, for example. And you can do all of this, generate the instructions. Now you go to the lab, you upload the instructions, and you can run your experiments. And when you do, and you generate some data, then that data is structured back to the design. And you can immediately actually have a quick look at what your response looks like across the different factors that you've specified. And then you can jump into the actual analysis of your data. No pun intended there. But you can see your raw data. You can identify outliers and exclude them if you think you need to. You can transform your data, doing a log transformation, for example. And once you're happy with it, you can move to finding the effects, building models. You can build multiple models and explore multiple models at the same time and actually see what the results of your DOE are. So in this case here, for example, we're seeing that glycerol and yeast extract have a positive effect, and you see how your response actually looks like when plotted against these two variables here. And so you can do all of this, essentially go through the entire DOE process within the same software platform.
That's really cool. Thank you, Nuno. I think for me personally, there's always something very satisfying about making that little switch over into DOE mode and toggling things on and knowing that the right things at the right point in my process are definitely going to be planned in accordance with my DOE. It's always a little bit of a nice moment to click that, make that switch over.
Yeah, it really enables you to make your experiment bigger and more powerful, right? Because if you need to check things one by one, then do you want to do 384 runs? Do you want to do 15,000 runs? No, but if you can trust that all these things are happening automatically for you, then yeah, you can go ahead and think bigger with your experimentation.
Yeah, absolutely. And for any of the automation scientists in the room, not having to do fewer water runs is always a nice bonus as well, right? Having that in silico. Cool. Okay, I'm a little bit aware of time, so let's keep going through the questions then. So another one for you, Nuno. How does Synthace support DOE adoption? So maybe for people who are newer to DOE or newer to automation.
Yeah. So I've been one of those people, and there's just a lot of new language, a lot of new things that you need to worry about, right? When we're thinking about DOE, then there's all these different types of factors and plots and designs that you might not be familiar with, and this can stop you from going ahead and actually just run your first DOE and learning, getting these amazing results out of it, right? And we took that into consideration when we built Synthace DOE. So we put all the things that people consider when they're running a DOE through this lens of biological relevance and automation compatibility, right? So then it's a more streamlined approach that is very context aware when it comes to biology, which should mean that it's easier for a scientist coming to it for the first time to adopt. But we know that there's more of, it's not just an issue with the tooling that you're using, right? There's ultimately, we're talking about people who are busy and we're asking them to change often the way they plan experiments and behave. So they need to learn new mental models. And this requires time that often people don't have, right? Or access to experts that can help. They need to gain proficiency to learn a little bit more about how and when and how to best implement. And also, once you're convinced, like, how do you make sure that you can scale across your group, your department, and you support the people that you're trying to get to use DOE going forward, right? And this is something where Synthace, the company, kind of also helps with, right? We have a dedicated support team, so people like Ross, if you're a customer, you'll have a dedicated customer success manager and field application scientist. And really, they're happy to talk to you of all things, from the stats to the automation strategy to the actual science that you're running, and of course, how to do it using our software. But also, we have quite a lot of documentation around DOE, explaining the stats principles, always using biological examples from those principles to how to implement it using DOE, step-by-step guides. And finally, we also have in-platform chat support. So you could be building your DOE in Synthace and share it with us and immediately go into a conversation with a member of our team that's going to help you. So if you're lucky, you might get someone like Ross. If you're less lucky, you might get me, but Ross sits just 2 desks down from me, so I'll just ask him, and within minutes, you'll have a good answer to your question. But Ross, actually, so we've been talking about a lot of the challenges about using DOE. So is it actually worth it, right? Is it worth the effort? How much time can you actually save, especially considering how much time you might spend planning the experiment in the first place?
Yeah, I think this is a really great question, actually. I think we did touch on it in one of the previous sessions, but I think it's worth revisiting. I think, yeah, DOE, as you've mentioned there, Nuno, it is ultimately like changing the way you think about and plan your experiments. And it can take a little bit more careful thought for the factors you want to screen, the things that you want to test. But ultimately, I think for us at Synthace, and for many of our users as well, maybe we're a bit biased, to be honest, but it's pretty much a no-brainer, rather than, I guess, me waxing lyrical about DOE, which for anyone who knows me will tell you it's hard to get me to stop talking about it sometimes. I thought I'd just share some of the value that we've seen from our users recently. So there's this lovely example here from Uncommon, who are a cultured meat company based in the UK. We had fairly close collaboration with them, particularly with one of their scientists, Alex. And I think the numbers here really speak for themselves. So they're looking at doing media optimization, lots of different sort of media components. They're looking at screening 22 factors altogether. And they were able to shave months off of their sort of media optimization timelines without having to really do any of the low-level nitty-gritty calculations, which Alex was very happy about. Saved him a lot of time in Excel. That's all something we can do less of. And then maybe on the slightly other end of the spectrum, looking at some of our customers in the pharmaceutical space, they've really seen a great sort of value in being able to do these sort of bigger, more complex experiments. There's a really nice quote here that the sort of Synthace platform enables you to run experiments that were previously impossible, which I think says it all, really. If you're able to execute a massive and these guys really are pushing the limits of DOE designs, big complex experiment, and ultimately drive time saving, cost saving, and improvement in the experiments and the scientific results that you're able to achieve. That's fantastic. And then along a similar line, but maybe a slightly different slant to it, with some customers recently at GSK who have a really interesting way that they tend to use Synthace, and I'll touch on this in a couple of slides as well, or where basically they will have usually industrial placement students will come in, and one of the first things that they do is sit them down with us and we take them through the platform and we train them, and really you'd be blown away by some of the science that they're able to do. And with DOE, it's really quite exciting. So yeah, there's an example here from Kizzy Gape, who presented recently a poster and actually was in our office presenting this internally as well. A lot of us were very excited to hear about what she'd been up to, where she was looking at assay development for a calcium ion flux assay and was really able to do some quite impressive things. It's an undergraduate student, early career researcher doing research that I think most PhD students, postdocs, even professors would be proud to have their name to. And similarly, we had another student sort of working the same time as Kizzy Cortha, who was working on sort of some safety assays for PROTAC development and was able to run an entire assay development in one single 384 well plate with a dragonfly. So shout out to SPT for that one as well. But yeah, really quite amazing stuff. So to maybe answer the question, is DOE worth it? I guess maybe I'd reflect that back on people and say, can you afford not to do it, right? You are able to do better science faster. I think that's something that we're all striving for in the industry, right? So
yeah, I think that's really on point. I think it's great when we see people like Thor and Kizzy like share, and often when they come into our office and share a lot of the detail that they usually wouldn't share outside, it's really cool to see just the depth and the size of the experiments they get to do and the sort of results they get within such a small period. But sometimes, yeah, there's a challenge of like, how do we actually get it to stick for a scientist, right? Or if you're trying to convince someone who's more reluctant at first, how do you actually get DOE experiments to stick?
Yeah, another great question. And this is certainly something that can be challenging. It's hard to demonstrate something to somebody without them having been through it themselves in a way, right? I think DOE can sometimes feel a little bit like a cult. There's a lot of people who do get quite fanatical about it and guilty as charged. I think, so we did actually a webinar on this a couple of years ago, I think it was, on DOE adoption and change management. So I've shamelessly borrowed some slides from that one, I'll be honest. But I think there's still good advice, as good as it was back then. I think there's maybe a few sort of top tips that I'd give people. One is that, first off, DOE, you're already deciding to take the leap, if you like, and you're keen to give it a go, get stuck in, get in the lab, try it out. It's pretty robust as a process. You can do a lot of things with it, make a lot of mistakes, if you want to call them that, and still ultimately learn and get things out of these powerful experiments. It is a robust process. There's also a lot of support out there to help you as well. If I was to say, I guess my next sort of bit of advice, and this is something that I maybe learned the hard way when I first started doing DOEs, is we'll always encourage people to take a little bit of a step back from the process that they're optimizing, ask themselves questions about it, and particularly when it comes to factor selection, it can be very easy to go in with predetermined ideas of the factors that you want to look at and the things that you think are biologically relevant. So we always try to encourage people to include things that they maybe wouldn't otherwise consider to be important. And particularly when you are able to look at interactions between factors, often that's where the biologically interesting stuff happens, is when you're looking at something you know is probably going to impact your system and something that you think won't. But this can soon balloon and you're suddenly looking at a list of 20, 30, 40 factors, which is fine, but maybe not something to do for your first ever DOE. Yeah, I think this sort of, to highlight this, we like, think big, start small. So it's okay to have those big plans, start with something simple, get to know the process, get to know what it's like to work with the platform or with the automation. You may be new to that as well. And you can always learn what it's like to go through those DOE iterations and take things from there. And share your journey. This is maybe a slightly corny way of saying, like, communicate, over communicate, both when planning your DOE, always advisable to, particularly again, to speak to that factor selection stage, to try and get the input from your colleagues, from people around you. Maybe they have some dogmatic experience or something that they've seen. Biology can often feel a bit mysterious at times. There's usually a reason. So talk to people, hear all those myths and legends about the system you're working with and include them in your, and ultimately share your successes. I think really the way to achieve that larger scale, longer stickiness with DOE, if you like, is to ultimately shout it from the rooftops, make it well known that you've been successful with something. So yeah, communicate relentlessly. And then finally, so this was something from Lauren in the previous webinar that I mentioned, know your why. So I guess to maybe dig into this one a little bit more, I really do think it is the most important aspect of this. There are lots of reasons why you could be doing DOE. Maybe it's to screen factors, maybe it's to optimize something, maybe it's simply just to understand a system that you're new to, but it's really important to work out like why it is that you're doing that DOE. It's ultimately going to be the thing that's going to guide you through the decisions that you make as you iterate, as you learn about your system. Is why am I doing this? You're going to unlock, hopefully, a load of knowledge about your system. And as curious biologists, it's very easy to want to follow all of those tangents. But I think really defining exactly what it is you're looking to get out of it can help you efficiently navigate through everything. And then just as an example of kind of what this can look like when we combine knowing our why with communicating, ultimately that change management side of things is not just at an individual level, it's ultimately an organizational level as well. It can be really important to make sure you're messaging that why effectively. So just a little example here of really inspiring people with this, right? It is a new and exciting thing for a lot of people to be doing and learning. And so yeah, don't be afraid to communicate that. And one of the ways I guess we see can help people to nail down this goal, this why, is to prioritize. So take a list of all of the possible reasons or reasons for doing that DOE or embarking on that optimization journey that you're on, and throw half of them away immediately. Figure out which ones are going to be most important to you. Take then the top 2 or top 3 maybe, and these are going to become your main objectives. The things from the second stage are maybe things that are a bit more like secondary. Yeah, you want to learn about them, but they're not the most critical outcomes. And then everything else is a nice to have. If you learn it along the way, then great. And if you don't, then don't worry too much about it. I think fundamentally as well, the real key to stickiness is not just learning the thing and doing it, but it's actually managing the cultural aspects of it as well. So just to reiterate a couple of the points there, these are the things that we tend to see across our customer base, that when people take this route, it really does lead to that sort of longer term, stickier change that you're looking for. So communicate relentlessly, as I've said, share your experiences, successes and failures, communicate with maybe other people in your organization who are doing DOE, even if it's on something completely unrelated scientifically. You can often learn from them. Creating ownership, really giving your scientists in the lab that time to learn this new way of thinking and new way of doing things. And then finally, I guess, ask for help. That can be internal resources, whether that's a stats department, if you're lucky enough to have one, or as Nuno mentioned, just contacting us here at Synthace as well. It's literally my job to help you guys do this stuff, and we love answering your questions.
Yeah, I think those are really excellent points. We all know change is hard, whatever it is, but it's also exciting, right? And I think we should, I think that communication of sharing all your successes is really important, because it really goes a long way in helping other people at least become more curious and wanting to try. And also letting people know that they have the space to try new things, right?
Yeah,
I think that's really important across any change in an organization.
Yeah, absolutely. Okay. So then I guess maybe changing tack ever so slightly here, we've got a little bit more of a technical question for you, Nuno, on the implementation aspects within Synthace, just around what sort of designs the Synthace platform supports.
Yeah, so as I mentioned earlier on, we try to think of what are the main sort of considerations are relevant for biology, right? And so we offer biologists 3 three different types of designs. So space-fill design, a optimal design, or a full factorial design. So a space-fill design is a design where you've specified your factors and your levels, and you're sampling between those levels evenly. And this is a really exciting way of exploring your experimental space. You have, especially if you don't know much about it, you really gain a lot of information, and you have an idea if your response surface is smooth or rough, or you learn a lot about a specific process. However, it's a bit less powerful and can be quite intensive when it comes to execution. An optimal design is often my default idea of a DOE, because it's that kind of crystallization that you can explore multiple factors at the same time, but doing it in a very efficient way, right? It's really boiling down to like, I need this number of combinations, and that's gonna be quite powerful at letting me identify if a specific factor has an effect on my experiment or not. And these are often used in the context of DOE campaigns, and you can really drive an optimization process using an optimal design. And then your full factorial design is essentially a combination of all the factors and levels that you define for your experiment. So it's incredibly powerful, but because it scales with the number of runs, then it very quickly becomes unfeasible to run. So really it's for a small number of factors and levels that you might want to do a full factorial design. But actually in many of those cases, maybe you don't need a full factorial design. But I'll let Ross talk about when you might test one or the other. I was just saying, so these are the main three options that Synthace gives you, but we know that there's people out there that are always doing wonderful niche experiments. And so we also give you the ability of uploading designs from third-party tools. And so you can still use Synthace to define your factors. If you're using a tool like Jump, you can export a JSL file, so you don't need to specify your factors both in Synthace and in Jump. You can export them from Synthace, upload to Jump, and save some time there. And then when you've designed your design using Jump or Design Experts, then you can upload it to Synthace, and we'll match it to the factors that we had already specified. And now you can continue your journey in Synthace, where we'll implement that design onto a plan that you can execute in the lab. And since we're on this, Ross, this is the kind of thing that if you're unfamiliar with DOE at first, you'll have this question: when should I use a space-fill design versus an optimal design versus a full factorial design or something else?
Yeah, thanks, Nuno. Yeah, we get this question a lot. I think it's often the thing that people ponder over the most. Is this sort of, it can feel like a very important decision, I think, to make, of which design do I pick? I've got my factors. How do I know which design to choose? We do have some functionality in the platform to help you assess the power of a design and give you some insight into whether it's appropriate or not to pick space-fill, optimal, full factorial. But I guess maybe just to give some high-level ideas of when you might want to use each of these different types of design. Really fundamentally, the first thing to understand when making this decision is like, what do I know about the system that I'm working with already? This is typically the thing that is going to make this decision, guide you in this decision. What resources do you have? So typically things like space-fill designs are a bit more of a shotgun approach, if you will. You're going to need higher run numbers to get lots of statistical power to look at things like interactions. But if you're just trying to understand a system from the start, then you don't maybe need to understand those interactions right away. So knowing where you are in your understanding of a system can be really helpful to make this choice. But yeah, generally speaking, it's about picking the right tool for the job, if you like. So space fills we look at as a way of exploring the landscape. So let's say you have a new process, a new assay, nothing about it at all. You've got these factors, these components in some mixtures that you want to test, but you don't really know where to put the levels. You don't really know anything about the system. They can be a great way of getting like a low resolution snapshot of what that design looks like. I agree with you completely as well, though, Nuno, on optimal designs. They typically be my first port of call. They're very flexible. They can be set up to be incredibly efficient. They're really the Swiss Army knife of the DOE design world. They're also great at handling things like categorical factors, which is something that's very common in biological systems. And then we have full factorial designs. Sometimes you've got the resources, you can do your massive test every combination of every factor, but yeah, they can be pretty expensive to run and not great if you've got more than 2 to 4 factors typically.
Yeah, I think if you're in a position to run a full factorial, then you're like, great, don't need to worry about it. But yeah, you're often not in that position, right? We always need to compromise.
Yeah, definitely. I think they can be, for people maybe running a DOE for the first time, it's often quite comforting to know that you are testing all of those combinations of things. And if there is some magical combination in there, that you're going to find it. And what we generally see is people will run them to start with, just build some confidence, and then from there tend to explore some of the more run efficient designs. Not sure what happened with our slides there. I think we both tried to progress it at the same time, Nuno. Yeah, so then next question for you, Nuno. If you could just maybe talk us through a little bit how Synthace handles categorical factors and also hard to change factors as well. I know these can be particularly, as I've just mentioned, particularly common in biological systems.
Yeah, we get this question a lot. Usually people think of a factor, we often think of numerical factors. So the concentration of a specific liquid that varies between two levels, but categorical factors are quite common as well. And these are factors that you can group into different categories, right? Like the name says. An example would be, I want to test 3 different buffers in my assay. Or you want to know if it makes a difference to get reagent X from supplier A or supplier B. So, for example, when we're thinking about buffers, you have, let's say, well, this. For example, when it comes to diluents, you just want to top up to a final target volume to make sure that all your components are reaching the target concentration. And so you're not thinking about this diluent, the concentration of the diluent itself. It doesn't really make sense to think about in those terms, but you want to say if having this diluent versus a different one makes a difference, right? And that's a very common example of a categorical factor. Sometimes we also end up in a situation where you want to compare 2 different liquids to each other, but you also want to vary their concentration. So in this case, we are adding a specific component at different concentrations. And these are what we call categorical factors. Mutual exclusion factors. So a very common example is, imagine you're running an enzymatic assay optimization. You're going to need to have some salt in there. You're going to have detergent and the reducing agent, but you don't know which one it might be best. So when it comes to salt, for example, you might want to test whether it makes a difference for you to use sodium chloride or potassium chloride. But then you also want to test the concentration of those salts. But whenever you use one, you don't want to use the other in the same run, right? Never both at the same time. And that's exactly what a mutual exclusion factor lets you do. And so if we look in this specific case at our final matrix, you can see that, for example, in run 1, we're using sodium chloride, and so potassium chloride is set to 0. But in run 8, we're using potassium chloride and not sodium chloride. Right? And similarly with the detergents. So Synthace will automatically know that these factors are in a way linked, and so that when you use one, you can't use the other. This is something that is quite common, and so that we introduced a way for the user to easily specify this type of experimentation. Now, another case that is also quite common in biology are these hard to change factors, right? So statistics or a key assumption behind these is that each run is independent from the next. But how often can you actually do this when you're running an experiment in the lab, right? Is it always possible to fully randomize your plates? And the answer is no. A very common case in biology is temperature. We know that temperature is key for all biological processes. And so often you might want to vary temperature slightly and see which one gives your best response. But usually, or it's very rare that you can actually control temperature at a per well level. You have a plate, and each well in your plate is going to have a different combination of factors, but it's very rare that you'll be able to heat each well individually. And so you need to split your runs on by plate. So each plate can individually go to a different incubator that is set at different temperatures. But now you've created the situation where you have this effect that is just due to your blocking of your experiment in a plate that is now confounded with the fact that each plate actually also represents an experimental factor that you're exploring, right? And so if we're in a situation like this, we need to say that this factor is a hard to change factor, because this will ensure that replication is added to each of the different blocks that you've created, so that you can discern the effect of the hard to change factor from the effect of splitting your runs into different plates. And so you end up with what's known as a split plot design. And this is, again, one of those cases where your ideal design kind of meets the reality of the constraints that come with an execution, right? And we need to go back to the design and adapt it so that it is still sound and you can still execute it. And to do that in Synthace, when you're specifying your factor, you can just say, hey, this is a hard to change factor. You tick that box and the design is modified to account for this situation. So similarly, and I think this is the last question, which is good because we're coming to time. Sometimes we don't necessarily have we need more than one well to get to our response, right? So a common situation in biology is you need to calculate signal to background ratios. So how do you do a signal to background ratio calculation if that is your DOE response, Ross?
Yeah, great question. Maybe to caveat the answer I'm about to give, I guess this is the solution we're going to talk about in a second is useful when looking at signal to background ratios, but also a whole sort of array of other different situations as well. As you've said, Nuno, where I need multiple measurements of some sample in order to give me the response that I'm actually interested in. And this can often be a challenge with designs that you're maybe generating in other tools, is that they're not set up with this in mind, and there's often no easy way to achieve that, and particularly if you're working in a tool that you're not super familiar with. And so yeah, when your response isn't a single measured value, how do you handle that? So to maybe take that example of measuring signal to background, you can imagine for something like assay development, this is something you run into very commonly, where you're looking at improving that assay window of the thing that you're trying to optimize. So what we really need is something that looks like this. So for every run in our design, we have one well that we're going to add maybe just a buffer to, and then a second well that we're going to add our compound of interest, or whatever it is that we're looking to test. And so the challenge here is that no single measured value actually represents the response that we're looking for, right? Whereas what I really want to be able to do is actually look at these two measurements for what I'm calling run one or condition one, and calculate the ratio between them, so I can then get that signal to background. And so in Synthace, we make this super easy for you. You can just define a factor within the sort of interface, specify that this is what we call a quasi-replicate factor. And what this is going to do is take your entire design, and as you can see on let me just get my pointer up, on the right-hand side here, we basically add a column into this design that we're going to call our quasi-replicate, and this will be replicating the entire design, taking into account the fact that this additional treatment is going to happen. This is a special type of factor where we're not using it to inform how the design is generated, we're just replicating everything against it. When we then come to look at that for all the runs in our design, it means we always have the equivalent with compound and without compound measurements that we can then use to generate the response that we're actually interested in. And as I've mentioned, this can be really useful for a wide array of different use cases. Both we're looking at things like signal to noise, assay window, like maybe direct comparison between different cell types, where we want to ensure we always have that comparative set of treatment within different groups of our design. And maybe even simple things like biological replication as well, where we're wanting to run the whole thing in pure triplicate and calculate things like averages from that as well. And so yeah, in Synthace, we make this super easy. As I've mentioned, you can specify a factor as being a quasi-replicate. And then when it then comes to doing the data analysis, we just simply pivot the data that you generate against that replicated treatment factor. and then provide some tooling for you to perform any sort of downstream calculations that you need to do to derive that new response that you're actually interested in from your data.
Okay, thanks so much, guys. Sorry, I do appreciate we're pretty much just on time. I wonder if we just have time to squeeze in one quick question. So yeah, we had a really good one. When you were talking about DOE adoption, some of the audience asked, like, how can you hit the ground running as quickly as possible? I think a sentiment we can all agree with. So you've got any thoughts, any wisdom to share?
Yeah, really great question. I think ultimately that's what me and my team are here to enable you to do. So we will work to train you on the platform, sort of with the basics of how to use Synthace, and then ultimately we're here to enable you to run your first DOE really as quickly as we can. We'll provide support with things like scoping your DOE, like understanding what factors you might want to look at, how to translate that into something that Synthace understands, so building your workflow ultimately. Maybe more generally, I think pick a system you know well, pick some factors that you maybe know are going to be interesting to investigate. Often we see people will quite successfully re-optimize something with DOE that they've maybe optimized previously without it. So you know what you're looking for in a way, and you can confirm then and believe the things that the DOE tells you. And that's generally a really nice way to sort of build that confidence in the process as well. So.
Yeah, I would definitely echo that, considering that if you're new to DOE and you come and you have a lot of questions around the designs and the tools and how you evaluate it, the one thing you're probably very familiar with is your system, right? And that is so critical for the success of your DOEs, picking the right factors and picking the right levels. And really no one is better placed than you if you're the expert in that specific area, right? And so leaning into that knowledge, I think is super important, really critical. And then it's about implementing it, and that's where what the tools are here for, and that's where we can help and support as well, right? But just leaning into the knowledge that you already have, I think, is really powerful.
Yeah, and maybe just to add one last thing as well, is that it's very easy to fall into analysis paralysis as well. As I mentioned, DOE is pretty robust. You can do maybe not the, like, perfect is the enemy of good in a way. Sometimes just giving it a go and figuring it out and learning is really the best way to get started.
Fantastic. Yeah, I think that the other thing that you mentioned, which really helps here, is know your why. Be really clear of the why. Why are you doing these things? Because that's the best guide, right? And it prevents those sorts of problems like analysis paralysis.
Yeah, absolutely.
All right. Okay. Sadly, I always want these things to go on a bit longer, which is very unusual for a webinar, but here we are at the end. So yeah, just a final couple of notes. First off, thanks so much to Nuno and Ross for all that amazing stuff. Brilliant, really enjoyable, lots to digest there. And yeah, if you're still hungry for more DOE content, we do have our DOE email course, which gives you a little bite-sized chunk about DOE every day for a few days. Yeah, it'd be great for you to subscribe to. And if you're still not satisfied and want to go even deeper still, we've got an 11-part series of videos of content covering all stages of the process and all these aspects we've discussed and much, much more. Yeah, particularly one of the most recent ones, how anyone can run DOEs with Nuno and Marcus, our wonderful CSO. So yeah, all available on demand on the website. Please do go and check that out. And yeah, very finally, do please fill in the feedback form if you want us to do more of this kind of stuff and make it more relevant and interesting to you. Just tell us what you want. But yeah, that's about it from us for today. So thanks very much, everybody. Look forward to seeing you again sometime. Cheers.
Bye, everyone. Thank you.